Hello, everyone. I'm Jerry Cole. I'm a stat biologist at the Institute for Bird Populations, and I'm going to be talking about evaluating and optimizing a comprehensive multi-species bird sound classifier, aka taking acoustic data and turning it into bird species classifications. My co-authors are Nichole Michel from the National Audubon Society, Shane Emerson from California State Parks, and Rodney Siegel, the executive director of IBP. So just a little introduction about what is a sound classifier. Sound classifier is essentially an algorithm that takes acoustic data and turns it into classifications, in this case, bird classifications. So we start at step 0. We go out there and collect acoustic data using an autonomous recording unit, an ARU. And then typically that data gets turned into a spectrogram, so a visual representation of the acoustic data. That's what step 1 is there. And then that's passed to a black box classifier, which then spits out an identification or a species it thinks is in that spectrogram. And then in this case, it's a California scrub jay, and then it usually gives a confidence score along with that of how confident it is that that is the species that's in that recording. So why test out this tech now? So one main reason is that we came across this excellent technology called birdnet, which is an algorithm that can identify over 900 bird species in North America and Europe, which is pretty amazing. Up until this point, most bird sound classifiers were done on a single species basis. You know, you had a species of concern, you built your classifier, and that was that. And none of these were really put together. But birdnet, since the Cornell Lab of Ornithology and Chemnitz University of Technology have such resources at their disposal, they were able to create this multi-species classifier that can take acoustic data and process it and give identifications for any of these species. So knowing all of that information, we thought it would be great to see if we can apply this to understand what's in our given study area, because with such a large number of species, any bird vocalization would get picked up and would be able to be identified by birdnet. Another piece to this is that processing is inexpensive and pretty fast. I'm sure all of you know all of these huge tech companies, Google, Amazon, Microsoft, they all have a cloud services arm of their company. And what you can do is take this birdnet software, put it into the cloud, feed in your acoustic data, and then get out text files that have bird classifications for your recordings, which is pretty amazing. You can do all this while you're sleeping, while you're working, and then come back to your data when you're ready and not have to have your computer clogged up with the program running. So another piece of this is we're interested in scaling up biodiversity monitoring efforts, especially in light of climate change. We have many bird species that are out on the landscape and in many areas that can't really get monitored that well or easily. So using this technology, we could collect acoustic data and quickly process it and identify, hey, these are spots that could potentially be suffering from some sort of loss of this given species, and we could take efforts to correct that. Another piece of this is autonomous recording units have decreased rapidly in cost. So for instance, the wildlife acoustics units, which are sort of the top of the line ARU units, run anywhere around $800 versus a little bit more inexpensive unit, which is the Audio Moth, which runs around $60. So what don't we know? BirdNet hasn't really been used for systematic acoustic surveys, at least in the peer-reviewed literature. We haven't found any studies that have used BirdNet for that purpose. And one of the main reasons is probably because it's so recent. So given that, we thought, hey, maybe we would try it out with our data set. We don't really know the duration of sampling that's needed to match the performance of a human annotator. So how long does BirdNet need to listen to a recording before it can actually pick up the same number of species as a human annotator can? So of course, we have the machine versus the human here. And then we don't really have any methods to maximize the data that's generated by BirdNet. BirdNet's great at pumping out all of these annotations, but what do you do with that once you're finished? We have these a lot of great methodologies that have been developed for in-person surveys of birds, but for surveys that are done autonomously and automatically by systems like BirdNet, there's a little bit less research, although that field is really super active currently, and we hope to see more methods that might come out to maximize this sort of data. And of course, another piece is the performance of birdnet with different ARU models, a cheaper model versus a more expensive model. So objectives, similar list, the duration of time that we need to accumulate the same species as a human, and how do we maximize birdnet's capabilities? So how do we get the maximum number of true positives and minimize the false negatives? So if a species is there and birdnet doesn't pick it up, how can we mitigate that? For this study, we used wildlife acoustic SM4 ARUs that sampled from sunrise to 11 a.m. We put these ARUs out in grassland, oak woodland, and chaparral habitat locations at a park named Carnegie SVRA near Livermore, California. That's in the San Francisco Bay Area, in case you aren't familiar. And then we took 10-minute segments from these recordings, these super long recordings, and had them annotated by a human. I'm the person who would annotate them. And then that same recording was annotated by birdnet, and also the full recording was actually annotated by birdnet. So I'm going to go a little sidetrack here on a primer on the terminology that we are going to use in this talk. There's two main terms, precision and recall. So precision is defined as true positives divided by true positives plus false positives. What that means is it's giving us a metric of, is the classifier too liberal with its identifications? So for instance, is it saying this particular piece of acoustic data is a given species when it is not? So does it have too many false positives? And then the inverse of that is recall. So true positives divided by true positives plus false negatives. So is the classifier being too conservative, and is it missing a lot of species that are indeed the species of interest, but maybe the acoustic data is too weak for birdnet to figure out that's what it is? So we're trying to balance these two things. We usually aren't able to achieve perfect precision and perfect recall, just given our settings, because these two things are sort of fighting with each other a lot of the times. But this is a little example of it below. So for instance, we have precision and recall are both 1 when we get all true positives in our data. And then another alternative is this big black box where we have lower precision of 0.3. So precision can range from 0 to 1. 1 is the best, 0 is the worst. So this says really low precision because we have a lot of false positives in there, species that look very similar to our target species, but are in fact not. So this is just a rundown of our methodology. So we have a 10-minute sound file per site. I annotated that 10-minute sound file that's on the left-hand side here, indicated whether I heard a given species in each of these 1-minute segments. If the species was detected on at least one of those segments, then we went on, collapse this data down to say, yeah, that species was there during these 10 minutes, at least once during this 10-minute period. And we put the same data through birdnet to allow birdnet to automatically annotate all of those data points. What birdnet does is it takes the 10-minute recording file, chops it into 3-second intervals, and then gives us a list of species it thinks are there and a confidence level for each of those species. We used a formula from Belantic and Donovan, a paper that does something similar with classifier data, and took all of these confidence levels and put them together to get an aggregate number of confidence for each site for that 10-minute period. These are the numbers for how confident birdnet is that that species is there during that 10-minute period. What we do is filter this down by a confidence threshold. We have to define, do we really believe that birdnet detected the species? So we'll filter out these lower confidence levels. So in this instance, this example here, we're going to say anything that's below 0.5 confidence level, we're going to say is not actually there, and anything that's above it is actually there. And then we have these data points here, down here for birdnet that we can compare to the human. We can get true positives, false positives, true negatives, and false negatives, and do get some statistics for precision and recall like we were talking about before. So the results. We were able to process recordings from 34 sampling sites. The human annotator detected 50 bird species across all of those sites. And birdnet had about 71% of the species right when we filtered at a relatively stringent confidence threshold. So you can see defining the threshold level for confidence score is pretty important here. So on the lower confidence score, we have a lot of incorrect species that birdnet came up with, but when we filter down those data to the ones that birdnet's very confident about, BirdNet does okay. 71% of the species were correct, and that's at least compared to me. So, I could have missed some too, and that's something we could probably dig into a little bit more in the future. So this is an output for precision and recall across a bunch of different confidence thresholds. So we're sort of working through all these different confidence thresholds, filtering out the data to see, can we maximize precision and recall at a given confidence threshold. And the way that works is we change this number. So this number, filter by confidence threshold, we change that to be a range of different numbers, either from 0.1 to up to 0.9, and then we get then we get a variety of outputs here, and we see how that affects precision and recall. For these 13 focal species that we're talking about here, that we had enough data to actually figure out precision and recall effectively, we see that precision is pretty good for many of these species. For some of these species, it's 1 for even for really low confidence thresholds. So even when we're being very liberal with the data. But another thing that we should also notice is that recall is not so good for many of these species. So about half of the time that the species was there at the site, BirdNet didn't pick it up, which shouldn't be that surprising, because acoustic data from ARUs oftentimes is not as easily identified by a machine as it is by a human. So a human probably could pick out, hey, maybe that is an acorn woodpecker that's far off, versus BirdNet might just throw it out because, hey, it's a very weak recording and I don't really know what that is. So how do we deal with these false negatives? One of the ways that we decided what might be good is to increase the duration of sampling period at the site. So listen to a longer recording. And given that, we can actually have a really stringent threshold if we wanted to, and it doesn't make any difference to BirdNet. It takes a negligible amount of time to process a longer recording, and then we can actually identify the species at a larger proportion of sites. So that's what these plots are. The number 1 on the y-axis is indicating 100% of the sites that a human said the species was there. BirdNet now also says it is there. And on the x-axis, that's duration of sampling period. So how long of a recording did BirdNet have to listen to get to that point? So a fair amount of these species, you have to listen to a pretty long, segment of recording. So maybe 200 minutes or 290 minutes, something like that. Some species it's shorter, but really this is pretty. So I said about a lot of these other things, but really this is for me pretty heartening. We can actually mitigate this problem of low recall for BirdNet. So when we apply this to all of the species, so this is using a similar method of filtering the data at different thresholds for all of the species, not just our 13 focal species, we see that the proportion of species that BirdNet is actually able to detect that a human said was there gets pretty close to, nearing 100%. Most of these sort of max out at around 90%. But to get near 100%, you might need a better BirdNet or a better acoustic sampling for the ARUs. And I think you probably may not get there, because I think the effective sampling radius for these units is smaller. Sorry, I should say is smaller than what a human's ability to hear a species is, at least right now. So BirdNet, I think, has the potential to supplement human annotation, maybe to take over that aspect of bird surveys. Not to say that you don't need humans still in the mix for this. We still need humans to verify that the species is there, to be able to make sure that BirdNet's doing its job appropriately. We see that BirdNet approaches human-like performance after about 5 hours of sampling. So one approach to using BirdNet for acoustic surveys is to choose a threshold that maximizes the number of true positives at the expense of having a lot of false negatives. So missing the species when it's actually there. But I think that's okay. I think it's good. It's probably best in this case, since we can leave the ARU out there longer, that's not a problem, and we can process more of the data. That we only want the classifications where BirdNet is very confident that that is what is actually there. And we can mitigate that problem of false negatives by listening to a longer duration of recording. I'd like to thank Tara from Carnegie SVRA for helping me with the study design and access to the park, giving me a lot of great ideas for how to accomplish this project. logistical assistance from Carnegie SRA staff, and funding from the Off-Highway and Motor Vehicle Recreation Division of California State Parks. All right, thank you very much.